antibiotic resistance. Early vaccine development and microbe discoveries. We begin in the late 1700s with Edward Jenner, who used cowpox to protect against smallpox, a process now called vaccination. Then comes Louis Pasteur, who didn't just use nature, he altered microbes by passing them through animals. Pasteur also proved that microbes cause disease, giving rise to germ theory. Next, Joseph Lister introduced disinfectants in surgery, laying the groundwork for sterile medical procedures. Finally, in the 1920s, Alexander Fleming discovered penicillin, the world's first antibiotic, from mold. It was a game changer, although it wasn't mass-produced until the 1950s, largely due to World War II delays. Antibiotics, discovery and impact. Penicillin was followed by streptomycin and others during the golden age of antibiotics. These drugs selectively target bacteria, sparing human cells. However, overuse has led to antibiotic resistance, a major modern concern. Branches of microbiology. Microbiology isn't one size fits all. It's divided into key areas. Medical microbiology focuses on diseases. Microbial physiology studies how microbes work. Environmental microbiology examines microbes in nature. Industrial microbiology uses microbes to create products like acids and bioplastics. Food microbiology investigates spoilage and safety. Understanding the microbiota. Let's zoom in on microbiota, the community of microbes living on and inside us. These are mostly beneficial or harmless. Normal flora aid in digestion, immune development, and even behavior. Mice raised in germ-free environments showed social and immune deficits, proving their importance. Emerging diseases and resistance. Historically, diseases like plague and leprosy spread easily. Today, HIV, Ebola, and West Nile virus are spreading faster than ever due to travel and globalization. Meanwhile, antibiotic resistance is rising fast. If left unchecked, infectious diseases may surpass cancer and death toll by 2050. Better diagnostics, new tools. Microbiology needs faster diagnostics. Rapid tests like COVID-19 antigen swabs are models for future infection detection. The goal? Create instant tests for all pathogens. Classification of life organisms fall into 3 domains: bacteria, prokaryotes; archaea, prokaryotes; eukarya, everything from fungi to humans. Viruses? They're non-living. No cells, no metabolism, but still infectious. Hack strains and scientific naming. We classify organisms using genus species like Escherichia coli. Strains are subtypes. For example, E. coli has helpful and harmful versions. In labs, we use safe strains like non-pathogenic Staphylococcus aureus to avoid risks. Part 1. Summary. Microbiology has come a long way, from smallpox prevention to fighting antibiotic resistance. With multiple specialties and advancing tools, it's one of the most vital sciences for human health. Review questions, Part 1. 1. Who is considered the father of vaccination? 2. Which scientist proved microbes cause disease? 3. What is microbiota and why is it important? Name 2 modern challenges in microbiology. 5. Are viruses considered living? Why or why not? Microbiology Lecture Part 2, Chemistry for Life. The chemical foundation of microbiology. Let's dive into the chemical basics of life. We start with atoms, which consist of protons, neutrons, and electrons. Each element has an atomic number equals of protons, an atomic mass equals protons, neutrons. Bonds, ionic, covalent, and hydrogen. Ionic bonds formed when electrons are transferred. Example, sodium Na donates an electron to chlorine, Cl, forms NaCl, table salt. Covalent bonds happen when atoms share electrons. Example, carbon and hydrogen in methane, CH score. Hydrogen bonds, weak but crucial. These bonds form between hydrogen and oxygen or nitrogen. They give water its unique properties and influence protein folding and DNA structure. R. Water, life's essential molecule. 70% of your body and 90% of microbes are water. It's a universal solvent that helps regulate body temperature and facilitates chemical reactions. Hydrophilic molecules dissolve in water. Hydrophobic molecules do not. Essential elements of life. Life is built from 6 main elements. 1. Carbon. 2. Hydrogen. 3. Oxygen. 4. Nitrogen. 5. Sulfur. 6. Phosphorus. Microbiologists must provide these in culture media for microbes to survive. Organic molecules, the stuff of life. Life is based on organic molecules, which contain carbon, the big four. 1. Proteins, made of amino acids, build structures and carry out functions. 2. Carbohydrates, made of sugars, store energy and provide protection. 3. Lipids, fats, make membranes and store energy. 4. Nucleic acids, DNA and RNA, store genetic information. Monomers and polymers. Monomers equals small units, e.g. glucose amino acids. Polymers equals long chains, e.g. starch, proteins. Covalent bonds link monomers into macromolecules, which are essential to all life forms. Searching for life on other planets. When scientists look for life, they search for water, organic molecules. If we find amino acids or lipids on Mars, that could hint at microbial life. Part 2 summary. Life's chemistry centers around atoms, bonds, and organic molecules. Understanding water, proteins, carbohydrates, and lipids helps us grasp how life functions and how to find it elsewhere in the universe. Review questions. Part 2.1. What type of bond involves shared electrons? 2. What is the role of hydrogen bonds in biology? 3. List the 6 essential elements in living organisms. 4. What are the 4 main types of organic molecules? 5. What's the difference between a monomer and a polymer? Let me know if you want part 3, organic molecules and DNA